This procedure uses live cell imaging to visualize the transfer of GFP-labeled HIV across virological synapses between HIV-expressing JurCat T cells and uninfected primary T cells. The cell-to-cell transfer assay starts with transfecting JurCat T cells with HIV-GAG-IGFP, an infectious fluorescent clone of HIV-1. The transfected JurCat cells expressing HIV-GAG-IGFP are then mixed with CMTMR-labeled uninfected primary T cells and loaded into an imaging chamber, where the transfer of GFP-labeled HIV across the virological synapse can be observed. The resulting data are analyzed using image analysis software to reveal the details of HIV transfer across T-cell virological synapses.
Hi, my name is Benjamin Dale from the laboratory of Ben Chen in the Department of Medicine at the Mount Sinai School of Medicine.
My name is Greg McNerney, under the laboratory of Dr. Thomas Huser at the NSF Center for Biophotonics at the UC Davis Medical Center.
And I'm Deanna Thompson, also from the lab of Dr. Thomas Huser.
Today we will show you a procedure for visualizing cell-to-cell transfer of HIV-1 using live confocal microscopy.
In this procedure, JurCat cells are transfected with an infectious molecular clone of HIV. Only trained laboratory personnel working in certified Biosafety Level 2+ or Biosafety Level 3 tissue culture rooms can carry out the protocol shown here. Working with infectious HIV in imaging facilities must be approved by the local institutional biosafety office. Maintain the JurCat cells to be used as donor cells at a concentration between 2 × 10⁵ and 8 × 10⁵ cells per milliliter in JurCat.